Good evening, everyone. Thank you for coming. My name is Chris Gobler, and I'm a professor at the School of Marine and Atmospheric Sciences at Stony Brook University. It's my pleasure to welcome you to the State of the Bays Symposium. Thank you for your interest in what we're doing here. Thank you for seeing the student posters. And the title of the presentation for 2025 is State of the Bays 2025. Keep calm and carry on. And as I think, probably everybody's aware, that slogan, of course, comes out of England during World War II. And we're not quite at wartime right now, but I think, depending on your perspective, sometimes and some days it might feel like it. So that's the slogan. It'll come up again later in the talk. I should acknowledge, firstly, thank you everybody for coming and your interest in our local coastal ecosystems. I do see Legislator Ann Welker here, so thank you, Legislator Welker, for coming. We have many of the Southampton Town trustees. I see Scott Horwitz, Matt Parsons, Chip Marin here. I may have missed some, but thank you for coming. And we even have East Hampton Town trustees here as well. So I thank all of you for your interest and for and your partnership as well and collaboration. And of course, I thank the fine young men and women who work in my laboratory. They are literally, yes, please. They're the boots on the ground and the fins in the water that make it all happen. And so we're here because of them. I'm dedicating this presentation, I don't want to get choked up, but to a couple of people who we lost too early, unfortunately, in the last year. So I'm dedicating this to Professor Bruce Bronowell. He was a great environmentalist, did incredible work. All across the world, really, but specifically lived on the East End, did a lot of great work in many important coastal ecosystems, made incredible discoveries, mentored incredible scientists who are now around the world, and was a great mentor to me. When I was a grad student, I took classes with Bruce, and through my running of the Center for Clean Water Technology, he was really, in many ways, both a mentor and a right-hand man who I could turn to when I needed advice and direction and just a really good opinion. So it was just a great pleasure to work with him. And I'll acknowledge his wife, Ann McElroy, who's here. And we all dearly miss Bruce. And then Craig Shione, who was a member of our lab, who was just an incredible young man. He had a disease by which he was beset with cancer literally his whole life. So he had childhood cancer. He passed away this winter. He was 33. But let me just tell you something about Craig. He was an incredible individual. He got his undergraduate degree through SoMAS. To show you how much he loved marine science, when he had cancer at the age of 5 and he had to be isolated, the toys he chose to bring into literally his bubble were little toy marine animals like sharks and turtles and whales. So when he finished his degree, the last thing he needed to do was get some final credits. He did it in our lab. And in the lab, and thereafter he did such an incredible job. I hired him to be a laboratory technician. And anybody who worked with him can attest to this, but he was a jack of all trades. He would do absolutely everything. And honestly, I only learned about his medical conditions after he passed, or at least the details of them. He knew all along that he was not going to be with us that long. And he lived his life that way. He every day showed up and he was so thrilled that he was a marine scientist. And he was diving in the water, no matter how dark and muddy that water was. If there was or how cold the water was, he was the first one to put on his wetsuit, put on the scuba tank, and go in the water. And he became this asset where people would actually argue, no, I want Craig with us today. We need him. So he was just so great and such a light. I have the word there, dragon warrior. He had an obsession with dragons, and I think it's because he saw dragons as big, powerful creatures. That's something that he wasn't in his life. He was never large in stature, but he was enormous in spirit. So we'll miss him greatly, and we're dedicated to both of these wonderful individuals, and they'll inspire us for years to come. Okay, so let's get to the subject of the day and talk about water, and the whole idea that we live on this kind of small island where water is at the center of everything that we do. We have a sole source aquifer, and therefore anything that we're doing on land is affecting our source of drinking water. And of course, what we don't extract to drink discharges into surface waters and affects those coastal ecosystems. And we know over time, as the population of Long Island has grown, the concentrations of nitrogen in that groundwater have also increased. And there's been more than a half a dozen studies done or more over the years as to why that nitrogen is increasing. We know that most of the nitrogen in Suffolk County in groundwater is coming from wastewater. Fertilizer is also very important, and on average is the second most important source. In some places, it can be as important as wastewater. But those nitrogen levels are rising. And if we look at where we stand with regards to the concentrations of nitrogen in our groundwater, and we look at what most of the country has, so if you look at that y-axis, that's 200 million people. Most people have relatively low nitrogen in their groundwater. So we're sort of at the tail end. And if you look at sort of a heat map, you can see there's some areas that are really high in groundwater, in nitrate in groundwater. Now, I'm going to spend this evening talking about effects on coastal water bodies, but I just want to briefly talk about potential health risks of that nitrogen, not to coastal ecosystems, but really just to humans. We know that there's already a standard for nitrate in drinking water, 10 milligrams per liter. But there's been a lot of research in recent years looking at the potential effects of nitrate in groundwater and human health. And this is just looking of human health, the links to cancer. And you can see this growth curve here. This shows you this is a hot research topic. And so the World Health Organization has classified nitrate as a probable carcinogen. And here are studies that are looking at the links between nitrate in drinking water and potential epidemiological links to cancer. And this is at levels below that 10 milligrams per liter. And I highlighted the ones that came out in the last year, 2024. There were a lot of studies that came out in the last year. There's also links to potential adverse birth outcomes. Some medical scientists that I know think that the links there might be stronger, but there's definitely not as much research on it. Just want to highlight this one paper and then a second one. This one came out last year from Denmark. Denmark is a small country. I didn't realize how small, just 6 million people. So just 127 cases of colon cancer per year. But in this particular study, they looked at what's the cost of reducing nitrate in drinking water to 4 milligrams per liter, which is, epidemiologically looks safe, and what would be the health system or public health benefit with regards to reduced medical costs from doing that. And they saw a benefit of over $300 million, which is very similar. Of course, the magnitudes are different to a study that came out of Duke University just about 5 years ago that, of course, more cancer rates, higher lumber because we have a bigger population here, but a savings of up to $1.5 billion in healthcare costs. And it's in parallel to all this happening. I will say that the EPA started a process of reevaluating what the standards should be for nitrate. Now, I will note that that process, has been slow. It started in 2017. It re-kicked off again in 2023, and where it goes from here is not clear, but it's something the EPA is looking at. Now, as I talk about this, and you think about nitrate in drinking water, you might think, well, I'll just drink bottled water, right? That's always an option. Just going to very briefly say that, of course, the other water issue that's emerging is microplastics. Right? And this study was an independent study. They don't really reveal the brands, but 3 different studies, all of them with 3 different brands of water, found that there's 250,000 microplastic particles per liter of bottled water. And this might have medical effects as well, right? And this is an emerging field, but this is a good paper and a good journal by very good scientists. So the thing I want to say is that we've got a drinking water source, and I'm going to talk about nitrogen in marine systems. But what I want to say is that having low nitrogen in our groundwater and in our drinking water is an optimal outcome, both for human health, but also for environmental health. Okay, so now moving into those marine water bodies, this is the map that we put together each year. We put it together at the end of the season, and it depicts where we saw hypoxia and harmful algal blooms across Long Island in 2023. So essentially spring through fall, and you can see we had more than 24 blue-green algal blooms we'll talk about, many low oxygen zones and many marine harmful algal blooms, and some fish kills overlapping with the areas of low oxygen. And I'll start by talking out about harmful algal blooms. We have last year and continue to have the types that make biotoxins, and also other types that are what are known as ecosystem disruptive. So they can lead to the adverse outcomes for either shellfish or finfish. And the research is in, we've done many studies where when there's more nitrogen, these organisms either grow faster, or in some cases, they can make more toxins. So I'll start by talking about the freshwater harmful algal blooms, also known as blue-green algal blooms, cyanobacteria blooms. And you're looking here at Lake Agawam, right in the village of Southampton. And you can see the contrast with the blue ocean water. So Lake Agawam and other freshwater bodies are often experiencing known as microcystis blooms. It's a particular genera of cyanobacteria that makes the compound microcystin. It's a gastrointestinal toxin, and that toxin can have adverse health outcomes. And these events are powerful when you see, for example, there's been studies out of Africa showing that there was an event in 2020, and actually a conservation area for elephants, where the elephants expired drinking water from ponds that had blue-green algae. This was a little bit of detective work. When it first started happening, people weren't sure, but what they're able to do is actually use satellite imagery to specifically detect the blue-green algae. And an unfortunate thing here is they actually were, in this case, keeping the elephants in a specific area to protect them from hunting. But that was an area that, because of a cycle of droughts and heavy rain, the ponds ended up having this unusual event. Beyond large mammals, our small mammal pets can be threatened by these events. These are studies by the CDC and the EPA over the years showing that that dogs can be poisoned by these compounds. And that's why the DEC has a very active program monitoring these events all across New York State. And this is what the data was for 2024. There's a lot of bars here, but a few things to highlight. New York State has 62 counties. 56 of the 62 experienced at least one of these events in 2024. But the interesting thing for us is, one, Suffolk County led the charge. So there are more of these events in Suffolk County than any other county in New York State. The other interesting thing is that, if you probably can't see it, but I can tell you, and you'll have to take my word for it, Nassau County is not up there. Right. And so, one interesting contrast is Nassau County is 70% sewered, and on Long Island, there's a lot, in Suffolk County, it's 30% sewered. And another difference there is agriculture, and some legacy agriculture was also a contributor. So this is mainly, obviously, a freshwater body system, but there is something that's been on our radar that is starting to come into focus for us, and that is specifically the potential for these toxins to go from freshwater into marine systems. The first landmark study on this was 15 years ago, where there was the anomalous death of sea otters in Monterey Bay. It was hard to explain. People weren't sure what was going on. You can see lakes with very dense blooms here occurring in this watershed and flushing out into this system. And just so you can see what you're looking at, this is what a normal liver looks like. And this is what a liver looks like that's been, essentially has cirrhosis. So bloody liver disease. So there was something affecting the liver. Microcystin is a hepatotoxin. And there were high levels found in the bivalves. And of course, when I first read the study, this happens to me all the time. You know, someone who studies smaller things, when someone asks me what a bigger thing is, like, what does a sea otter eat? My answer was not clams, but the answer is often clams. And so that was the route of exposure here. And so the overall idea is that these the blue-green algae and or their toxins can flush out into marine ecosystems, potentially get into shellfish. We have looked at this recently, and we've looked at many systems, actually from the Chesapeake all the way up to New York. These are the systems we've looked at in New York. We've published a couple of papers on this. Marcella Wallace is here, and she did her master's thesis on this. She has a great poster on it outside, and the second top paper there was just published in the last couple of weeks. This is happening in New York, and I'm going to leave it to Marcella to give you some of the details on that. But we do know that nutrients and specifically nitrogen can play a role in these events as well, making them both more intense. But there's the microcystin compound. It's a nitrogen-rich compound. And so because it's nitrogen-rich, more nitrogen available for the organism allows it to make more toxin in the cell. Moving to marine ecosystems, recently we was putting a lot of time into looking at Dinophysis acuminata. This is a dinoflagellate that makes this compound here, okadaic acid, that can lead to something known as diuretic shellfish poisoning. We find this organism everywhere, in fact, all times of year, usually at low levels. But here's a map of where we found the density we found at different locations, and you can see there are some hot spots. I mentioned there's at least 2 of the East Hampton Town trustees here. Did I miss any of them? I know. Yeah, okay. For the East Hampton Town trustees, I've been monitoring their coastal waters for the past 11 years, and that's allowed us to put together a really nice data set to look at changes over time. And so this is one organism that we've noticed in the last few years is becoming more abundant. And it's actually popping up all across the US. So if I was giving this talk 10 years ago, 15 years ago, I would say we've never had a DSP event in the US. But since then, there's been events in the East Coast, the Gulf Coast, the West Coast. So there's a paper I was on with some colleagues from the East Coast, West Coast, and Gulf Coast at NOAA and other fine organizations, sort of documenting the most intense events that we've seen, the different species, strains, toxins. It's hard to see, but of all the locations, that box on the right is cell densities. The most intense event recorded in this paper, and therefore that we thought about in the US, was in New York. Last year in 2023, we were tipped off actually by a former student who said, have you seen Little Seatuck Cove? And I said, no, I've never even heard of Little Seatuck Cove. But he sent me this picture. Lucas Chen is here. He's not here. He left. Okay. He was here. He went and got water to find that this was not only did it look bad, but it was not 2 million cells per liter, which we thought was the highest amount, but at 100 million cells per liter. So really, really dense. The bloom went on, as you can see in the upper right, it's with a log scale, but at a very high level for more than a month. The DEC closed this area. It was already closed to shellfishing, but they added to it a biotoxin closure for things that could be harvested there. We collected shellfish, sent them to colleagues at VIMS, and indeed found that there were 400 nanograms per gram of the Dinophysis toxins. That's more than double the FDA limit, so the closure was a very good idea. So with a bloom that intense, we geared up in 2024 to go out there, really understand what's going on. Why would we see this anomalous bloom? So we got out there early in the season. We were detecting the fresh water or measuring what was going on with regards to fresh water and other things. Started monitoring in April, and instead of getting 100 million cells per liter, we just got 1,900 cells per liter. So very anomalous. There one year, there the next year, but certainly not at the same levels. But interestingly, just a few miles away, and sometimes science does feel like whack-a-mole. It just so happens we've been studying another site called Reeves Bay, which is the southern part of Flanders Bay, if you're not familiar with it, for a few years now. And we saw essentially fairly low densities in the prior years. 7,000's getting up there. But then suddenly, last June, we had another one of these enormous events. So it's 60 million cells per liter. That's what the water looked like. You can see the streaking of it there. This is what it looked like under a microscope. Suffice to say, we had to dilute that down a little bit to get an accurate count. But this is a bloom that built up through May, peaked in June at that very high level, but we had levels, that extremely high for almost a month in the system, making it one of the denser blooms that we've, only number 2 to what we saw in 2023. We've studied these over the years. These blooms are what are known as mixotrophic. I don't have time to go into the details of all of what that means, but because they both can act like plants and animals, essentially that's what it means. People had always been focused on that it's what they are eating that's more important than the nutrients, but we've definitely been able to show that here, particularly in the Northeast, that the nutrients gives this organism a boost. And Lucas isn't here, but his poster is out there. The organism grows much faster when there's high levels of nitrogen. And we've shown that recently. These are experiments from East Hampton at 3 Mile Harbor, where when we add nitrogen, it grows faster. And again, I'm calling Lucas out like 3 times now, and he's not here. So he's hopefully he'll see the video and he'll feel bad about it. But this no, I don't hope he feels bad about it. I hope he feels good that I'm calling him out. We've also tried to do the reverse process to say, well, what if we reduce the nutrients? What does that do? And so that's what's shown here, where we're reducing the nutrients by 50%, 75%, 90%. We have a control and this artificial seawater that has no nutrients, so diluted by those different percentages. And you can see, even at 50%, we got a little bit of reduction, but a big reduction, tenfold, when we finally hit like a threshold of 75%. So just saying that we're going to need some big nutrient reductions to limit these blooms. And frankly, it's consistent with what the Suffolk County Subwatershed Study has found. And many of these places going to need about a 60% or more reduction in nitrogen. And that nitrogen does make these events less intense. Moving away from harmful algal blooms, another water quality parameter we're very much focused on is oxygen. Right? Oxygen, of course, is needed for all life to exist, except for some classes of bacteria. It's one of the few parameters that, through the Clean Water Act, there's an actual standard that's set by the DEC. They want to see 4.8 milligrams per liter. They never want to see it go below 3. If we look at what we saw in 2024, we went below 3 in a lot of places. Now, this is partly because we now have sensors that are measuring the oxygen every 10 minutes, right? And so the upshot of this is what we've learned over the years is that this is what we call nocturnal hypoxia. The oxygen levels are getting low at night. The question is, does that matter? And you can guess what the answer is, but I just want to show you why it does matter. Firstly, digging up some old studies. Mike Dole was here. So this is the first showing us some of his results where he put out oysters in a few places where we knew the oxygen was good in a particular year and when it wasn't good. And the responses were different, but the oysters grew faster when there's more oxygen. So Jeff Kramer is here. He was here. And he designed some experiments. Maybe I should just stop calling out students as they keep getting this signal in the back. But he did a great master's thesis where he grew different types of bivalves, clams, oysters, scallops, right over here in Old Fort Pond, which experiences what I would call mild hypoxia. And he tried to fix it by running that water from the pond through this maze with lots and lots of bubbling. And he was successful. So this is what oxygen looks like normally. You can see the day-night cycles. The peak is during the day when there's lots of photosynthesis. The troughs are at night. And again, we're falling just below that standard. And so on the one hand, before having done this research, I might have said to myself, well, it's just a little below the standard and it's only for an hour or so each night. Maybe not a big deal. And again, he could fix that just by aeration, right? So keeping it always above that standard. What he found is that indeed the bivalves do grow faster when you give them more oxygen, just like we saw in the field. And that you can see the regression here, a very good relationship between the improvement of growth and how many hours of hypoxia they experience at night. The one weird thing about just experiments is that sometimes there was a strong effect from aeration, And sometimes there was no effect at all. And I couldn't figure it out until we looked at what was actually happening. So if you didn't have nighttime hypoxia, well, they didn't care if you gave more oxygen. But if there was a lot, the more oxygen made a big difference. The last thing that Jeff did is he actually then took all of our oxygen monitoring data and then used those regression models to go back and say, if the oxygen were higher at all the locations, how much would the bivalves be benefited? And you can see the percentages in some places are very high. And yeah, I put the dashed line showing that those sites to the right of the dashed line is similar to what we saw for Old Fort Pond, which is to say that, it's a fairly mild system. We have other systems where the hypoxia gets much more severe and the gains would be much higher, 100% up to a 200% improvement in growth. And that improved growth rate is important. Oh, there's Jeff. He is here. Great job, Jeff. And that improved growth is important because this is work that we've done where we put out different size bivalves, specifically clams, and just say, what's their survival rate based on their size? And the answer is if the clams are really tiny, none of them survive. So they need to grow fast to get to a threshold size in order to make sure that they don't perish. And unfortunately, sometimes a lot of the clams that are planted are these very small ones. Okay, shifting gears, and I want to talk about the Anthropocene, right? We're in close quarters here, so I can say the words climate change. I mean, it's funny on the one hand, but Mike could tell you about the meeting we had yesterday where we had to redact those words from a grant proposal. We were asked to. But we do need to think about it if we're going to manage coastal ecosystems. And the signs of climate change are everywhere, right? The temperatures in 2024 were the highest ever by 1.5 degrees centigrade. And that was despite the fact that the Pacific Ocean had transitioned to a climate phase called La Nina, which is an extreme cooling phase. So I sometimes call that Earth's air conditioner turned up as high as it can go, and we still set a record. And this is what it looked like across the planet. You can see some areas were really far beyond the normal. We had Earth's warmest day last summer. And the funny thing is this is June 21st, which shattered the record of June 20th, 2024. So back-to-back days that were the warmest. And this winter, of course, here, we might think, oh, well, thankfully we're back to normal. But did you know, January 2025, first you think back to how, that was a cold January. That was the warmest January planet Earth's ever had. Right. And again, despite the fact that we're in a La Nina phase, and you can see, I mean, just your eyes weren't deceiving you. Yes, we were actually we had a cold January, but the rest of the planet seemingly did not. And we know our coastal waters are warming. This is showing the warming trend just since 2002 on a per year basis and just in summer. And so our summer water temperatures are increasing. And when we have warmer water, in some cases, that can lead to more rainfall, more evaporation. And in fact, it's been documented here by NOAA that since the middle of the 20th century, the Northeast in particular has been experiencing a 71% increase in the amount of heavy rainfall. So these are like tropical deluges, the rainfall falling all at once. And if you're around last August, you may be familiar with the event that happened last August 19th. Now, the interesting thing about these summer rainfall events is that they are quite regional. So what you'll see, I see Dean Shepson, the Dean of SoMAS, also known as Shep here, nodding his head because he's in the Stony Brook area where they had more than 10 inches of rain. But interestingly, on the South Shore, due south of Stony Brook by not many miles, they had hardly any rain whatsoever. Here on the South Fork, we had about 4 inches of rain in total. But these rainfall events are impactful. Firstly, they closed down shellfish beds, over 3 inches of rain, and that will lead to that. And some of these places were closed down for an extended period of time. And there was a lot of damage. For those of you who might know the Stony Brook area, here is the roadway that goes past the Stony Brook Mill Pond, and that's what it looked like before. That's what it looked like the day after. because there was so much water that fell, the levee broke. And this is what the pond looked like before and after. It still looks like that today. The same thing happened for Blydenburg Lake, where this dammed up system, the dam broke and the water fell out. And the headlines for this event is that this was a 1 in 1,000 year rainfall event. And that's the headline there. And even the governor called it a 1 in 1,000 event. And as I read that, I thought, 1 in 1,000. Didn't we have something like this recently? And I do keep track of these things. So I went back to Hurricane Ida, and it was about 10 inches of rain, and it was only 3 years prior. But then I thought, but wait, what about that event before then? And I'm at that time in my life where it seems like it was just a few years ago, and then I found out actually it was a decade ago. But I found it, the 2020 to 2014 event, right? And again, I might be the only one in the room who remembers it, but 14 inches of rain fell in Islip in 24 hours. That's the record for all of New York State, right? And again, this was regional, and there were some areas that were much lower. You can see the prior record, if you can see it, was 11 and a half inches of rain. But there's the third event in a decade of over 10 inches. So I'm not sure these should be called 1 in 1,000 year events. And in fact, there's this study that had just come out looking at going forward, not what's happened, what might be happening with regards to the intensity, the frequency of these events. Whether there'll be 1 in 100 year events that are 1 hour in duration or 24 hours in duration. Either way, what you can see is this model predicts that we're in the bullseye for these events. And so the 1 in 100 year storms is predicting that maybe, 10 years or less. By my math, it's about every 3 years now. And we need to think about this because it's not just that dams break, but with more rainfall comes more nutrient loading. This is my colleague Anna Michalak. She's at Stanford. And this study essentially showed that this rainfall alone is going to increase nitrogen loading by more than 33%. Right, that's with us doing absolutely nothing. It's 33% more, and we need to account for that. And again, in her maps that she put in that paper, you can see right where we are and close to the Midwest, we're in the bullseye. Okay, a final climate change thing I want to talk about is how organisms in the ocean move as oceans warm. And so that's a sort of a central tenet of climate change biology. Organisms want to stay in their thermal optimum. Water's warm. To stay in that same temperature they were in, they have to migrate north or south, depending on the hemisphere they're in. So one of these organisms that we need to think about is something called Vibrio vulnificus. Here's a bacteria that loves warm water. And what you see here is over time, through an annual season, the incidences of infections with Vibrio vulnificus, I'll talk about what it is momentarily, and temperatures. So it's temperatures in both Florida and Texas, and cases in those places as well. And you can see they track pretty well. So this is a bacteria that seems to like warm water, brackish water, can infect open wounds, and the immunocompromised people are the most vulnerable, but other people can be infected as well. I think we have a young person here who thankfully is with us here, but had a pretty bad infection of Vibrio vulnificus, being exposed in East Hampton. And this, the organism really just eats through flesh. And so it's a scary thing, and the outcomes, are not great. So it's pretty serious. With Hurricane Helene and Milton last year, it triggered a bunch of these infections in the South. Now, something that caught my eye a couple of years ago was this paper, not even 2 years ago now, where it showed over time Vibrio that used to just be a Gulf of Mexico phenomenon migrate north and essentially move up the East Coast. And by the end of the study period that this paper looked at, had made it all the way to New Jersey. And then they did a model saying, based on what we know with regards to potential changes in temperature based on CO2 emissions and time of year, that potentially By the end of the century, it could migrate up into Long Island and into the Northeast. This paper was on the mark for about 3 or 4 months until actually in 2023, sort of in advance of what we thought, it had migrated to the Northeast already. I will say, having spoken to people, even though despite the fact that it does spread through open wounds and shellfish, that there's no indications of it being in shellfish in any of the cases here. And I will say there are very good protocols in place by the DEC for icing down shellfish as they're harvested. That was for another type of Vibrio called Vibrio parahemolyticus. That's also going to work well for Vibrio vulnificus. But at the same time this happened, the CDC put out an alert to doctors. This is not like for the public, but this is for doctors to be on the lookout for these events. This is 2023. We found the organism quite widespread in 2023, and we found some particular areas on the east end and in the North Shore that had fairly high levels. We found it in about a third of samples and about half of the sites. We were back at it again in 2024. We focused in on the hot spots. Levels were a little bit lower, but still fairly high. And in looking at the environmental trends, the two things that become very obvious is that, yeah, it likes warm water. It disappeared as soon as we hit a certain temperature threshold, 17 degrees or so, and was inversely correlated with salinity as well. So the highest levels in the warmest water and the lowest salinity waters. Now, something we always do in our lab is to try to understand, beyond looking at the conditions within the environment, is to try to do some experimental work. Is there something we can do to make it grow more or less? Can we combat it by changing things? So we did some experiments. We actually experimentally changed temperature and changed salinity. We couldn't see a very big response. But Ruthie Drinkwater, who's here, worked up the digital PCR assay for this organism, also did some experiments where we, thanks to people at the Center for Clean Water Technology, I'm not sure if they're still here, but got some wastewater, both what we would call raw wastewater and sand filtered wastewater, and ran an experiment, simply taking the water, letting it incubate on its own and exposing it to the raw or sand filtered wastewater. And we got an enormous response. I've been doing these experiments a long time. You've already seen some of these. If we see a parameter go up by like 50%, it's a lot. This is a 2 order of magnitude increase in 24 hours. So this wastewater is rocket fuel for this organism. And, Ruthie can, this is one experiment, but we sort of, again, we tested different things iteratively through the summer. And once we landed on this, it was just very, very consistent, essentially. So this Vibrio vulnificus, is sort of you can look at it as the combination of things. It's the warming, more rainfall is lowering salinities. And, there is a link there for the wastewater. Okay, so that's a lot. That's a lot for you to look at. And, you may be feeling like this dog here, but I want you to remember our motto for the day. We're going to keep calm and we're just going to carry on, because there is good news. And I do want to turn to that because there are great examples of where great efforts have been made to improve conditions and the outcomes have been great. So, for example, the Long Island Sound study that was put together by the EPA, right? They brought together New York State and Connecticut and said, what can we do to address what's known as the dead zone, this huge area in Long Island Sound of low oxygen? And they said, we're going to reduce nitrogen loading by 60% at significant cost for billions of dollars to upgrade sewage treatment plants. But what you're looking at here is what the dead zone looked like before and after. And you can see the dead zone is not gone, but there's this enormous part of Long Island Sound that no longer is experiencing this dead zone. So that's it in it aerially. But here's a plot of nitrogen loading into Long Island Sound and the size of the dead zone. And not my data, I'm just plotting the data from the Long Island Sound study. This is a pretty tight correlation. It's hard to find correlations that are this close in an ecosystem setting. Essentially saying we turned off the nitrogen and this dead zone got smaller and smaller and smaller to the point that in 2024, it was the smallest it's ever been at 43 square miles, which, there's certain areas that we think may have been hypoxic going back before even settlement because of this area by the Narrows. But this is one of, I would say, the great examples in this country of people saying, let's address nitrogen loading and affecting a great outcome. But it's happened in other places, in some cases sort of accidentally. What you're looking at here is the Bergen Point Sewage Treatment Plant, which collects wastewater from the Southwest Sewer District, the last major sewer district built in Suffolk County. And this plant went online and began sending wastewater out to the ocean in around 1981. Before that, the wastewater was going right at the Great South Bay. And what you can see here is a plot of the total nitrogen in the area near there. And, there's definitely seasonality and spread, but certainly a decline once this system went online. But more importantly, what I want to emphasize here is the incredible expansion of seagrass in this area once they got the sewage out of this region. An expansion of nearly 3,000 acres of seagrasses in this area as a before and after, right? And that's during the same period of time where other systems were losing seagrass. And then a final example I'll point to is in the Huntington Bay, Northport Bay area. This is an area that's experienced harmful algal blooms for many years, going all the way back to 2006. The epicenter for these blooms has always been in this area right in the back. We've mapped out what we call the cysts of this organism. It's always at the highest levels there. If you map out cell densities, they're at the peak here, and then they decrease sort of monotonically outward. Just so happens the village of Northport has a sewage treatment plant that discharges right there as well. Maybe not a great combination, but in 2013, they got funding to upgrade their sewage treatment plant and essentially to reduce the nitrogen loading from about 21 pounds a day down to less than 7 pounds per day, so a threefold decrease. So this is the acreage of that system that was closed to shellfishing each year before upgrading the sewage treatment plant. And it was pretty reliably a large area. You see on average, almost 6,000 acres. System upgraded in 2013, and it's been pretty different since then. The blooms are less intense, but certainly the shellfish bed closures have become pretty uncommon. Now, there's other things going on here. There's other nitrogen sources, but clearly this had a positive impact on the system. Okay, so coming now to the nitrogen loading in other parts of Long Island, and specifically here in Suffolk County, and for all of Long Island, if you add up the two counties, we've got about 400,000 homes that are discharged into ground. So what are we going to do about this? Well, there could be a solution, and it could be a political solution. Now, we know there was a big election in 2024. Elections have consequences. Now, we know in Suffolk County, President Trump won by 11%, and both of the U.S. representatives won by more than 10%, or more than 12%. On the same ballot, there was an initiative, Proposition 2, where the same voters who came to the same conclusion were posed with the question, do you want your taxes raised for clean water, right? Or no? Now, some people, if I would have told them the outcome before, might have said, well, maybe it would be a no. But in fact, not only was it a yes, this was an overwhelming yes. 72% of the citizens of Suffolk County said yes. Raise taxes so we can have clean water, right? I find that incredibly heartening, right? And that's an issue that everybody can agree upon. We all value water here in Suffolk County. And clearly, people voted with their heart and their conscience and their, that what do they want? They want clean water, something we can all agree on. So this, in an era where, maybe everything does seem like it's on fire, this is something to keep focused on, that we are in an era where everybody's in agreement. We all want clean water. Everybody's working together towards that. And it's very for me, very, very encouraging and important. I could show the statistics that New York is shrinking faster than any other state, but despite that fact, Suffolk County is still growing. So these initiatives are important. And Suffolk County has been an incredible leader in wastewater. I have the tagline from worst to first, because I can say with confidence, and particularly with the passage of Proposition 2, that there's no county in the nation that has a better plan for upgrading septic systems than Suffolk County. And they've got a very science-based plan on where that should get done. You see the hot pink areas, the 200,000 systems they seek to upgrade by 2050. And that's not a willy-nilly drawn line here. There's a lot of science that went from deciding, yes, let's protect these areas because they contribute to drinking water. Let's protect these particular water bodies because these are the most sensitive to heavy nitrogen loading. And other areas, you know what? We don't need to focus on those for right now because they're doing okay. So this was not made up haphazardly. And the difference in what, if we don't take action, and if we do, would be enormous. And I'll just emphasize, remember that 4 milligrams per liter for drinking water health. There's not much of that with the implementation of the watersheds plan. So there's many systems that can be installed to upgrade on-site septic systems. As you may know, I direct the New York State Center for Clean Water Technology at the university, where we're harnessing science to engineer clean water to protect public health and the environment. And we've created something we call nitrogen-removing biofilters that are very simply systems built on sand and wood chips. And they've been doing extremely well. So this is the nitrogen effluent for all systems. It's getting dark early here. Don't want to put anybody else back to sleep. But this is the nitrogen effluent for, as you can see, many systems that have been tested here in Suffolk County. And I'm excited to say that the system that was just approved by Suffolk County, the nitrogen removing biofilter box system, has achieved the lowest nitrogen effluent of any system in Suffolk County at 5.6 milligrams per liter during testing. And that's to the engineers having figured all that out. I'm not taking any credit for that. But that's important because these systems can make a big difference. In some cases, threefold better. I'm just going to show very briefly the way they work. They have a layer of sand that goes to a box filled with wood chips. And again, they're getting this incredibly low effluent. They're also leading to the removal of all sorts of drugs, pharmaceuticals, and personal care products at percentages that rival sewage treatment plants. And, with the passage of Proposition 2, and with some very progressive actions in East Hampton and Southampton town, homeowners can get up to $50,000 to upgrade their systems. And in many cases, if the installation is not too complex, that will cover both the installation and the full restoration of the property. And the nitrogen removing biofilters are available for those installs. Okay. A final thing I want to talk about is what we're going to do while we upgrade those systems. Upgrading the system is going to take time. The travel of groundwater takes time. And so I'm going to talk a little bit about specifically how kelp can help and talk about a little bit about the work we've done with seaweeds. Kelp, of course, is photosynthetic, takes up CO2, so it's combating some of our climate change issues, takes up nitrogen, producing oxygen, which is in short supply, and is active against harmful algal blooms. Mike Dole is here. He is the pioneer who brought kelp to Long Island. If you didn't see the article in the New York Times, full-page article calling him the Johnny Appleseed of kelp, because he literally has been spreading it to all over New York waters. There's Mike with our first site that the first site we worked on, Great Gun Waste Farm in Richards Bay. And so one of the things that we've been focused on is how can it affect ocean acidification. I don't have time to go into all the details, but suffice to say, high levels of CO2 acidifies seawater and can have ill effects for bivalves. And this is the most recent readings for CO2 in the atmosphere. For those of you keeping track, that's up, a lot. I'll just say that. 280 is where we were pre-industrial. So we've done experiments with kelp and oysters on that Great Gun oyster farm. When we do that, we put the kelp out and we map out pH. We see that the kelp creates this tongue of high pH water compared to the rest of the water there. That's in space. In time, we put out sensors. We see over time it's creating high pH water. And the oysters like the high pH water. These are growth rates of the oysters, whether they're grown on the kelp or near the kelp. And compared to a control site, they're doing much better when they're grown with the kelp. We also know that the kelp can actually deter harmful algal blooms. I haven't talked about Alexandria, but this is one of the most dangerous of the harmful algal blooms because it makes saxitoxin, a neurotoxin. But when this organism is exposed to kelp, this is what it looks like normally. And here's Alexandrium spilling its guts. Right? It does not like the kelp. The kelp has bioactive compounds that leads to the lysis of these cells. You can see it in the pictures, so you can see it in the data. Here's an experiment where we added nutrients, or now we can see the light, or we added kelp with nutrients, and you can see, a threefold decrease in the amount of Alexandrium with the addition of kelp. And so we're very interested in applying this in the field. And we've even had some results where we think there's even a third mechanism for helping bivalves. So gets rid of the harmful algal blooms, combats ocean acidification. Here's an experiment from my graduate student, Lane Silvers. And she did experiments where, yes, she showed that with S. lentissima is Saccharina lentissima, the sugar kelp, it can raise DO, it can raise the pH, both on average and over time. There's some wiggle here. But when we did this experiment, and well, I could say, and here's the growth of mussels, a more than doubling of the growth of mussels during the experiment. I mean, double. That's a big number. It's not 2 orders of magnitude, but it's still a big number for bivalves. But the thing about this experiment is that the oxygen levels were fine, even in the control. The pH levels were fine, even in the control. There were no harmful algal blooms. And so there's emerging evidence that actually the kelp itself is a food source for the bivalves. The kelp is sloughing off cells as it's growing and actually has a complement of fatty acids, for example, that are different than what's in the plankton. So it's almost like getting a more diverse diet for these bivalves. And so we think this is yet another reason why cultivating seaweeds with bivalves is a good idea. And we're exploring other seaweeds as well, some red seaweeds, as you can see here, and even ova to grow during the summer. If you didn't know, kelp is mainly a winter seaweed, but we're having some success in growing other types of seaweed to do the same sorts of things during summer months. And we call this, or I call this, the halo effect. We are not solving climate change with seaweeds. We are not going to get rid of harmful algal blooms with seaweeds, but they can be used in a targeted way on shellfish farms in specific areas to have positive outcomes for shellfish against ocean acidification and against harmful algal blooms. So I'm at the end. I've showed you that excessive nitrogen loading from wastewater is an ongoing threat to ecosystems and human health. Climate change is accelerating, and coupled with nitrogen is having a compounding negative effect on coastal ecosystems. Upgrading septics is the primary mechanism to reverse water quality impairment and to improve drinking water. But in the meanwhile, seaweeds hold the promise to regionally mitigate water quality impairment. So I thank you all for your attention. And I'd be happy to take any questions. Yes.
Wastewater treatment,
Yes.
they last.
We think they're going to last for decades, potentially centuries. And on the one hand, that might sound silly for me to say that, but these boxes are saturated, so they're filled with wastewater and they're anoxic, no oxygen whatsoever. Bacteria grow at 1/20th the rate under anoxic conditions compared to oxic conditions. So very slow degradation. And that's what they're meant to do, which is very slowly degrade, very slowly release carbon to promote the things happening. So we think the answer, I'll conservatively say decades. Yeah. But I'll also say one other thing. wood chip box, that design specifically is meant that the box can be accessed. So if for some reason, there's the thought that those wood chips, maybe they have gone bad, maybe things are getting clogged, they can be vacuumed out and replaced. Yes, sir.
You seen any effects from the gutting of the EPA by our old friend, Mr. Zeldman?
Well, no direct effects for me yet, but I will say that the New York State DEC, to my understanding, gets 1/3 of its funding from the EPA. And I've also heard that EPA budget is to be cut by 65%. So there's some math in there, and I have lots of state support. So I don't know what's going to happen, but some of the math there suggested that could be on offer, and there are things happening afoot that that are bound to have trickle down effects for sure. And I know some really good scientists, PhD scientists at the federal level, who have been doing really good work, who lost their jobs in the last month or so. Yes.
I know you've been working in East Hampton Village on Georgica Pond, and I was just wondering how that's going.
It's going well. Thank you for asking. Georgica Pond had the worst blue-green algae blooms in New York State in 2014 and 2015. And we made a series of changes for Georgica Pond, working on getting nitrogen and phosphorus out of the water and out of the watershed. And we have the current and former executive directors of the Friends of Georgica Pond here. And Sarah, how many septic systems have been upgraded in the watershed, would you say? I know the answer is dozens. I just don't know how many dozens.
Yeah, well, I mean, it's a loose number because we don't, when we count, which are underway or completed, but we're getting closer to 40, something like that.
Yeah. So there may be, it's a place that's had probably more upgrades per water on a water body than maybe anywhere else. So, and again, the system's been responding. So more work to be done, but positive things moving in the right direction. Anybody else?
We don't talk
A little bit about that problem, sorry.
Yeah,
Sorry, I was like, kept going.
go ahead.
Could you talk a little bit about that prop, and if there's proposition, and if there's any action tests in there? Who's tasked with that and is Stony Brook?
Yeah, so Proposition 2 had a couple of components to it. One is to make Suffolk County sort of be responsible for all the wastewater, so have a central management entity. I'm looking at Walt because I know he'll have the exact term. Yeah, central wastewater, single wastewater district for the entire county, but also importantly, it puts a 1/8 of a penny sales tax. So 1/8 of a penny ends up being 12 cents on $100 purchase, but it ends up being millions and millions of dollars, maybe even billions over decades, to help fund the upgrade of septic systems and sewage treatment plants. Yes.
It seems like our nitrogen levels for groundwater are okay. Is that what I'm reading? But is it necessary to have a reverse osmosis water system nowadays?
Right. So that's a great question. I think the most important thing is to know the nitrogen levels in your drinking water supply. Right. And so some people have Suffolk County Water Authority water, and you can get that data from Suffolk County Water Authority, which is very progressive. They start treating for nitrogen when the levels get above half of the MDL, so at 5, which is great. If you have private well that you want to get that tested, and if you have private water and you're above, say 4 or 5, you might consider putting that on there. I can say I had a private well for a few years for my second house that I had, and in a short period of time in testing, the levels went from 2 milligrams per liter to 4 to 8. And then thankfully I got public water and now much lower levels, but knowledge is power and testing is important. Anyone else?
One other question.
Sure.
It seems like the dinoflagellates, the toxic ones, seem to peak in the summer
Yes,
when we're supposed to go swimming in the bay.
yes.
Is that true for the ocean?
Ocean water is very clean in general. Everything, almost all of the problems that we see are in backwater areas. You know, there's like the two components back there, lack of water flushing and then the high levels of nitrogen. And together those things, you know, that's what leads to the dinoflagellate blooms because the water's not moving around. But out in the ocean, everything is better. Water is cooler, the salinity is higher, the algae are not there, the bacteria levels are low. So enjoy the ocean. Yes,
Because of that, and you said that
Sorry.
the ocean is warming, are we gonna have an effect on the Gulf Stream?
we are not here. But certainly the patterns of the Gulf Stream are changing. It's a little far afield, but yeah, it's a longer story. So yes.
I've mentioned 40 conversions in the Georgia Pond area, which is great. But your slide showed 200,000 conversions by 2050.
Yep.
We're not living in that world. How do we get from, the relative small numbers we're doing
Yeah.
on the South Fork to getting to 200,000 by 2050?
Yeah. Well, that's what Proposition 2 is meant to do. That it's meant to accelerate. And the Suffolk County Department of Health Services, which is the sort of the gatekeeper in this whole process, is ramping up towards that now. They're increasing staff. They're trying to streamline the process. And so that's why Proposition 2 happened. It's meant to make that happen and make those things accelerate.
But providing more money clearly isn't a problem for people around Georgia Pond and Lake Agawam. Proposition 2 and the incentives.
Yeah, but
that are being facilitated by the town of Southampton and the towns of East Hampton are about money, but clearly those populations don't lack for money. So what's the resistance? Why aren't they all just doing it right away?
well, I mean, you need to speak to each of the people in there, but I'll just say the advantage of the money for Suffolk County is more staff to make the process happen faster. And then, each homeowner has their own choice to make. I will say it is a extensive process.
So there's no sort of regulatory or legal incentive to force property owners ultimately.
New construction.
Yeah, new construction must have these systems. And if you expand your home more than 25%, I can't remember if that's in footprint or value, you also have to have an upgraded system. And there's been talk of, it hasn't happened yet, but there's been the next legislative boundary, if you will, maybe on sale of home, but that hasn't been implemented yet. There's been discussion of it.
That's what's happening on Shelter Island. If you purchase a home within and they don't have a nitrogen reducing system within 3 years, it's not set in stone, but that's what they're entertaining, that you would have to replace the system, and you can get, of course, you can get the grants to do that.
Chip, I'm going to make you the last question.
I got wrapped up in the blue-green algae of the students that were out there.
Okay, yeah.
I was noticing the correlation with the phosphorus, and it was interesting that it actually a reduction of the phosphorus levels made the algae itself more toxic, but the actual bloom levels were larger. So there's an interesting kind of like, or it's almost like a stress response of
Yes.
a weakened organism, just kind of like a plant can put out
Yep.
a stress response if it's weakened. You mentioned, obviously a lot of this was targeted towards nitrogen. The PRBs, what is the effects of phosphorus loads with PRBs? Because I know we'd like to start to implement PRBs in certain instances where there's road runoff.
Yeah. So just for everybody's Chip Marin, Southampton Town Trustees, asking about PRBs, permanently reactive barriers, which is a way to intercept groundwater And as the groundwater, and actually encourage groundwater to flow through a particular barrier, they're typically filled with wood chips or some other carbon source to promote the removal of nitrogen. There can be materials put in to absorb and remove phosphorus, although depending on the watershed, sometimes the phosphorus is already removed in the sandy sediment. So really, they can be modified to remove phosphorus, but the need to do that is really going to be location specific. Okay, well, I've been jabbing away for too long, so I want to thank all of you for coming tonight. Thank you for your attention. And if you're interested, of course, the students who haven't turned tail and left will be back at their posters if you want to talk some more science. So thank you and good night.